Good afternoon, everyone. My name is Abdulaziz Bashir Kutawa by name. I'm going to present on this very topic, antifungal effect of Khaya Senegalensis leaf extract on plant pathogenic fungi. These are the presentation outlines. In general, plants produce a remarkable diverse array of over 500,000 species of low molecular mass natural products. These natural products are also known as secondary metabolites. The medicinal value of these secondary metabolites is due to the presence of chemical substances that produce a definite physiological changes and action within an organism. The most important of these include glucosides, glycosides, alkaloids, fatty oils, flavonoids, and steroids. The screen of plant extract and natural products for antimicrobial activity has shown that higher plant represent a potential source of new anti-infective agent as well as serving drug discovery from natural products for primary late compounds. Khaya senegalensis, which is commonly known as mahogany, it is a popular medicinal plant in Africa, and this plant belongs to a family called Meliaceae. The rhizopus species have also emerged as an important cause of morbidity. Different species of fungi are responsible for destroying major agricultural commodities. This could be after harvest or before harvesting period. These products become contaminated by fungal growth and mycotoxins. Aflatoxins are still recognized as the most important mycotoxins. The expression of aflatoxin-related diseases is influenced by factors such as aging, nutrition, sex species, and the possibility of concurrent exposure to other toxins. The main target organ in mammals is the liver, so haplotoxicosis in humans include limited availability of food, environmental conditions that people the growth of molds on foodstuffs, and lack of some agencies that can regulate the aflatoxins monitoring and control. Therefore, this work will help in identifying the pathogens as well as determining the suitable concentration of the leaf extract that could be used to control the growth of plant pathogenic fungi. The objective of this study are: 1. To isolate and identify different species of fungi from spoiled tomato. 2. To determine the effect of different concentration of leaf extract of Khaya senegalensis on some plant pathogenic fungi. For samples collection, about 20 samples were randomly selected from 4 different locations. The locations include Hospital Road, Haingada, Kanangaru, and Isakaita Road. The leaf extract were prepared based on the method described by Bashir et al. 2018. For the isolation and identification of the pathogens, it was carried out based on the method of Tijan 2017. Then after isolation, uh, the colony or the culture uh, was subjected to different uh, series of repetitive subculture in order to obtain the pure culture of this fungi. The fungi were grown on a media called SDA, that's Severed Distress Agar, and incubated at a temperature of 27 plus or minus 1 degrees Celsius. For cultural and morphological characteristics, different characters like shape of the spore, color of the spore, color of the cornea, size of the conidia, and color of the colony were taken into consideration. The viewed organisms were compared to the standard reference keys, that's Atlas, for possible identification, and all the data of 10 were subjected to analysis of variance, that's one way ANOVA, using least significant difference, that's LSD. Based on these results of mycelial growth, the most suitable concentration was found to be 500 milligram per mil, which was having the mycelial growth of 1.42 mm and inhibitory percentage of 75.9%. The least concentration to inhibit the mycelial growth of this fungus was found to be 100 milligram per mil, with the mycelial growth of 2.91 and the inhibitory percentage of 50.6%. For the mycelial growth, the control was found to have the highest growth of 5.89. For the Aspergillus niger, the mycelial growth was also found to be highest in control, and the most suitable concentration to inhibit the mycelial growth was found to be 500 milligram per mil, with the mycelial growth of 1.60 and inhibitory percentage of 72.84%. The least concentration was found to be 100 milligram per mil, which was having the mycelial growth of 3.97 and inhibitory percentage of 32.60. The results further revealed that the antifungal activities of the extract were enhanced by increasing the concentration, that's from 100 to 500 milligram per mil. Hence, the inhibition activities of the extract were concentration dependent. These findings are in agreement with the report of these scientists who indicated that the increased antifungal activities correspond with the increase in the concentration of the plant extract. Khaya senegalensis possess a fungitoxic effect by inhibiting the mycelial growth reduction against these two species of fungi. The antifungal activity of Khaya senegalensis goes in line with the results of these very scientists who stated that the extract was effective to inhibit the growth of Fusarium moniliform and Aspergillus flavus. The fungitoxic properties of Khaya senegalensis could be attributed to the presence of some compounds like saporonins, alkaloids components, which has been identified as the antifungal agents in different plant species. The aqueous leaf extract of Khaya senegalensis exhibit an antifungal activity and could provide a therapeutic agent for the treatment of ailments caused by Rhizopus tulinepa and Aspergillus niger. The extract from leaf extract of Khaya senegalensis was found to be more effective on Rhizopus mycelia than in the case of Aspergillus niger mycelia, and the best concentration to inhibit the growth of this fungi was found to be 500 milligram per mil. Based on the conclusion, the tested plant extract can effectively control fungi that are causing some diseases in plant, and this makes it a potential fungicide for disease control, as the method is environmentally friendly and cheap, and it showed an effective fungicidal and fungitoxic potentiality. These are some of the references used during the study. Thank you so much for giving me your time. With this, I have come to the end of my presentation. Thank you once again.